Hello again. In this video, let's do a brief introduction to the central dogma of molecular biology, aka how we go from genetic information to something like a protein product. So the central dogma explains how we go from genotype, so all of the instructions encoded in our DNA, to a certain phenotype, so an actual physical characteristic or a function of an organism, like a human. Okay, so like I said, we're talking about flow of information. So DNA is the molecular structure that holds all of the genetic information, aka instructions, on how to be you, how to make you as a human. But again, how do we go from code to an actual physical, tangible thing? Well, DNA gets turned into, or I should say, DNA gets used as a template to make RNA, which is like a little tiny transcript. And that RNA gets used to make protein. That's like the really simple gist of it. But we're going to go just a little bit more in detail. So first off, I want to point out that when we're thinking about the central dogma and starting with DNA, we have to think about the fact that DNA undergoes a process called replication in certain scenarios. So this is thought of as part of the central dogma. Think of a scenario when DNA has to make more DNA. Okay, maybe pause, maybe think about it. But basically, anytime a cell needs to make a new cell, what will happen is there will be one primary cell. It's going to double everything inside of the cell. That means make more organelles, make more enzymes, make more DNA. And once we have double everything, we can, in a very organized fashion, divide everything and split the cell down the middle. And now I go from one cell to two cell. So this doesn't happen in every single cell in your body. For an example, your neurons can't do this. They can't make more neurons. Instead, it's mostly happening in your stem cells. So stem cells make more cells, which can go on to make more stem cells or differentiate into different cells like red blood cells, neurons, muscle tissue, etc. Okay, so DNA is sometimes replicated. And one of the interesting things to think about is like, how does that happen? So first, we need to remember the structure of DNA. So let's start with the nucleotide. This is the basic building block of DNA. So we have a phosphate group and a ribose pentose sugar, and that's going to be attached to a certain nitrogenous base. This is one nucleotide, and many nucleotides strung together make up one of the strands of DNA. But DNA is double-stranded, so you often see it drawn as this double helix. So when you see that double helix structure, there's one thing I want to point out. First off, this backbone of the DNA, like the side of the ladder, that is referencing the phosphate group and the deoxyribose sugar. And then we have these rungs of the ladder. And those rungs represent the nitrogenous base part. Okay, cool. Now the thing is, there are 4 different nitrogenous bases that can be found in a nucleotide. Maybe you remember, there's adenine, thymine, cytosine, and guanine. And here's the really cool thing. If you think about the human dictionary or like human alphabet, we can create really complicated dialogue and narratives just shuffling around, 26 letters in our alphabet. It's the same thing here, except the alphabet is much smaller. Instead of many letters, we just have 4 letters, but still, the way those letters are arranged, it spells out instructions for how to make certain proteins and thus make you. So that's why we care so much about this structure. So when you see these rungs in the ladder, it could be A, C, T, G, G, C, and so on. It's literally spelling out a code. Another thing to remember is that if we focus on the sugar, it helps us understand directionality of the nucleotide. So I didn't make this up, somebody did, but basically what you do is you count the carbons. So remember, in chemistry, anytime there's a point, that's an implied carbon. So the first carbon, going clockwise, is right here at that first point. Then there's the second carbon, third, fourth, and I didn't draw it, sorry, but there's a fifth one there. Okay, I was being a little bit lazy in my chemical structure. So just like me standing here, I have two ends to my body, right? This is the head, and then there's my feet. If I were to do a handstand, you'd be like, Kayla's upside down. You know that, right? Okay, so there's a directionality to these nucleotides, right? The phosphate group is up top. So the way that we reference that certain directionality is by counting these sugars. So wherever the 5 is sticking up, that's the 5' end of the molecule. And wherever the 3 is, that's going to be the 3' end. So if I was looking at this little ladder I built, maybe this is 5', maybe this is 3'. This becomes really important because when we're making new DNA or we're making RNA, it always gets built in a certain direction. So all of these enzymes always build 5' to 3', adding on to the 3' end. Another important thing that's going to come up is that DNA, and honestly, like any pairing in the nucleic acid world, is always anti-parallel. So I said one of these strands would be 5 to 3. That means the other strand is going to be running in the opposite direction, 3 to 5. So one goes 5 to 3, the other goes 3 to 5. So DNA is anti-parallel. And then the last thing we need to remember to understand how DNA replicates and also like how transcription works, which is our next step in gene expression, you have to remember the base pairing rules. So for our different bases, anytime I have an A, it always pairs with T. If I have a C, it always pairs with G, and vice versa. G always pairs with C, T with A. Okay. This applies for when we're making RNA, but the difference is RNA doesn't have thymine, it just has uracil. So anytime there should be a thymine, instead you put a uracil. Okay, so real quick for replication, let's say we have DNA, it's good, it's in its nice little double helix, and we need to make more DNA. The way this actually happens is through a process that's referred to as semi-conservative. Because we conserve that original DNA strand, but it's actually going to end up in both of the new strands. So let me show you what I mean. It doesn't happen all in one go, pulling the strands apart, but little by little, these strands are pulled apart. To make it easier, I'm going to act like the whole thing gets pulled apart at once. So here we go. I pull apart my double helix, right? So there's one side here going 5 to 3, the other side going 3 to 5. So I pulled it apart. An enzyme called helicase is going to help do that. And then to make the new strand, all we have to do is complementary base pair. So if I were to zoom in, and we can pretend that there's a certain sequence here. So maybe it's A, C, 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 G, A, T, and so on. An enzyme called DNA polymerase is going to go along and just use the bases that it sees to build the complementary strand. So anywhere there's an A, great, we're going to put a T. Anywhere there's a C is a G. G to C, A to T, and so on. And that will keep happening until we have an entirely new strand on both sides. So you can see how I took my original strand. This is like simplifying, but you pull it apart, use the side that you do have as a template to build the other strand of DNA. And that's why it's called semi-conservative, because I have 2 strands of DNA here. Half of the strand is from the old DNA, from my original cell. And then half is newly synthesized, so it's semi-conservative. Understanding this is really helpful for now when we get to talk about gene expression and transcription. So the first part of the central dogma we learned is just DNA makes more DNA anytime a cell needs to divide. But how do I go from DNA to protein? Because proteins have so many different functions in the human body. It helps explain things we can see like hair color, eye color, skin color, right? Those are the easy ones because you just see them with your eyeballs. But even things like personality or like cell signaling, things that you can't see with your eyes, a lot of it's just controlled by proteins and enzymes and basically the final products of gene expression. Okay, so I'm drawing this big circle here, and this is going to be the cell. For a eukaryotic cell, DNA is not just floating around. I guess technically neither for bacteria. In bacteria, it's in a region of the cell called the nucleoid region, but for eukarya, we have DNA being housed, let me make it a little bigger, in what's called the nucleus. So this is a membrane-bound organelle. And DNA is arranged into chromosomes. It'd be kind of crazy for me to draw it right now, but nucleus. But we have 46 chromosomes and they come in homologous pairs, so 23 homologous pairs. And if you were to zoom in on one of those chromosomes, we'd find that running along the length of all the chromosomes are little segments of coding regions called genes. And I'm going to oversimplify, but let's say one gene codes for one protein. It can be a little more complicated, but that's the standard. So one of these genes codes for a protein. Let's say it's a protein that helps make pigment in the eyeball. So like a brown eye pigment. So it has to do with eye color. I want to use the instructions in the DNA to build proteins. Here's the problem. Do you remember where ribosomes are found? Ribosomes are the machinery that builds protein. They're usually depicted as these tiny little circles, ribosomes. You can find them free-floating in the cell, and you can also find them dotted along the endoplasmic reticulum in the part that's called the rough ER because it's dotted with ribosomes. Side note, if the ribosomes are on the rough ER, that's usually because the protein that's created is going to go through a process of being processed and tagged for secretion outside of the cell through the endomembrane system. A lot of proteins that are made from ribosomes in the cytoplasm are just going to stay in the cell, so they're not going anywhere. Okay, so you're kind of seeing the problem here, right? Like instructions are in the nucleus. I need to get to the ribosome. It would be kind of bonkers to try to like take all of my DNA and send it to the ribosome. Also, the ribosome's too big to make it inside of the nucleus, so what are we going to do? We're going to make a small copy of the gene we want. So let's say that this is the gene of interest. We're going to copy down the gene in a small segment of nucleic acid called RNA. RNA is single-stranded, and this is messenger RNA because it's a messenger for the ribosome. So that process right there, maybe you recognize it, this is called transcription. And think about the word. When you transcribe something, like in English, you're writing it down word for word. So if I have a passage from the textbook and I'm going to transcribe it into my notes, I'm just writing it down word for word. So I'm taking the nucleic acid DNA and I'm creating a transcript, also in nucleic acid language, just in the form of RNA. That RNA is going to go through some processing steps inside of the nucleus, and that doesn't happen if you are a bacteria, but that's okay. We're staying in human world. And then it's ready to leave the nucleus and go find a ribosome. Amazing. So because this is meant to be an overview, I'm not going to go through all the steps or all the players involved. So really briefly, the ribosome, along with the help from tRNA carrying amino acids, is going to read that mRNA transcript and use it to build a polypeptide. So these green little circles that I'm drawing, this is going to be our polypeptide strand. And we know that polypeptides are made up of amino acids. You fold those amino acids up, you fold up the polypeptide, maybe there's multiple subunits, and you get a protein product. Dun, 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 dun. Okay, so this step of going from reading mRNA to making protein is called translation. Kind of making a bit of a messy drawing here. So here's how you can remember it. In translation, we are going from RNA, which is a nucleic acid, to protein. That's a different type of macromolecule. So we're switching macromolecule categories here. I'm translating, right? When I translate from English to Spanish, I'm switching languages. I am switching languages here from nucleic acid to protein. Okay, so now I have my protein product. If we were thinking about eye color, right? The cells in my eyes are the ones that are expressing this gene. And they are creating let's say it's a protein that's an enzyme that's going to help create brown pigment for my eyes. So you're seeing how we're going from genetic code to a physical manifestation of a certain trait or characteristic. Okay, I hope that gives us a good starting point. Next, you'll want to dive deeper into each of these steps individually.